Bismillah, Assalamualaikum warahmatullah. You're listening to Immunology lesson and this is part 2 of our discussion on antigen and antigen recognition molecules. We talked about B cell receptor or antibodies last time, so today we'll learn about T cell receptor or TCR. The first point is TCRs most often detect only short peptides. And this is in contrast to B cell receptors, which can detect more stuff, including peptides, but also intact folded proteins, nucleic acids, carbohydrates, lipids, and small chemical compounds. It doesn't mean T cells can't detect all these at all. They can, as you'll learn later. It's just most of the time we associate TCR with short type peptide recognition. You might notice by now that when you study our textbook, you see many names and it might confuse you because they did confuse me too back then. So when that happens, don't think it happens because you're not smart. I've been there before. Well, I'm actually experiencing it again now, but in a different field because I'm studying philosophy of science and Kalam, and in certain sense, it's more confusing than immunology because the technical concepts there, sometimes they use the same names, but their meanings are contested by different worldviews. For instance, secular versus Islamic worldviews. So my point is that confusing feeling is part of learning any complex disciplines. So you just need to be patient, and the more you study, those names will map out in your mind. So you would know that when you see T lymphocytes, you know that it's T cell. When you see CD4 positive T cells, it usually refers to the same thing as T helper cell. CD8 positive T cells, that means cytotoxic T lymphocytes or CTL. Now, it doesn't mean that T helper cells are the only ones that are CD4 positive or CTLs are the only ones that have CD8. No, they are cells that actually have both of them at the same time. So these are just how the CDs are most commonly used. CDs are short for cluster of differentiation. So these are naming conventions for molecules found on cell surface. We find them in proximity to our T cell receptor. The reason for that is co-stimulation. Well, let me explain it like this. So CDs are useful for at least two things. Number one, we use them for cell identification. So when you get a sample of unknown immune cells, you can check for CD profiles. So if they are, for example, CD4 positive, CD3 positive, CD45 positive, but CD8 negative, then you know that it is T helper cell. So that's number one. Number two, the reason we find CDs often close to T cell receptor like this is they play the role of co-receptors. It means that at times your T cell receptor binds to the antigen, but that binding alone is not enough to activate T cell. So to activate the T cell, you also need the CD molecules to bind at the same time. Now, what do the CDs bind to? They bind to MHC, major histocompatibility complex. So try to say that really fast. Major histocompatibility complex. Where do we find major histocompatibility complex? We find them on the surface of antigen-presenting cells. Antigen-presenting cells are cells that activate CD4 positive T cells. Examples of APCs are dendritic cells, who activate naive T cells to be CD4 positive, and macrophages and B cells, they stimulate the existing CD4 positive T cells. You're going to find also that these three cells, DCs, macrophages, B cells, you'll find them being called professional antigen-presenting cells because other cells that have nucleus can present antigen as well, but they're not considered as active or as professional as these three guys. These professional APCs, they do a couple of things. Their main job is to display peptide MHC complexes, or you can also say antigen MHC complexes. They also add stimulations to the interaction between APCs and T cells. They do that using the co-stimulators they have on their surface and by secreting specific cytokines. For example, you've got a DC as your APC and it encounters a T helper cell. On your T cell, you have T cell receptor and you have CD4 and other CDs as well, like CD28. On your APC, you have your antigen displayed by class 2 MHC. So firstly, your T cell receptor will bind to the antigen. But more often than not, that binding alone is not sufficient to activate your T cells. You need co-stimulation to verify. It's like the two-step authentication for our sensitive accounts. When you sign in to your crypto account, for example, and you key in your password correctly, you still can't log in. You need a second authentication. So the website will ask you to give the code that they send to your phone. And only after you put in the code, only then your login is successful. So these co-stimulators, co-receptors, and cytokines, they are like that code sent to your phone. They behave as secondary authentications. So when you have the T cell receptor binding properly and the co-stimulators binding correctly at the same time, only then you get a successful antigen presentation. And that process of antigen presentation, it is enhanced by exposure to microbial products rather than non-microbial products. And this principle is applied when you make a vaccine, for example. When you make a vaccine, sometimes you can add pieces of microbes to enhance vaccine performance. And that additional substance is called an adjuvant. T cells can also enhance the antigen presentation by sending signals back to the APC. So for instance, CD4 positive T cells use CD40 ligand and interferon gamma to bind to corresponding receptors on APC, and that binding will amplify the antigen presentation. A major APC in our immune system is the DC. You have classical DCs, two types of them. By the way, I think I mentioned major types of DCs briefly in one of my previous lectures. So now you learn what they do. CDC1, which is conventional dendritic cells type 1, focuses on ingested antigens, transferring them from the vesicles to cytosol, which is important for presentation of antigen to CD8 positive T cells. On the other hand, CDC2 presents antigens to CD4 positive T cells. Plasmacytoid DCs releases type 1 interferon, useful for viral infection. PDCs also take antigens in the blood and bring them to the spleen. For monocyte-derived DCs, they can be found in inflammatory conditions, but we're not sure what they do. Langerhans cells are found on epidermis and seem to work a lot with macrophages. Their function is suggested to be similar to CDC2, which is presenting antigen to CD4 positive T cells. Among all our APCs, CDCs have been shown to be one of the most efficient for primary T cell responses. So factors for this are, firstly, their strategic locations. So they are found, for example, on common entry sites and tissues where they can catch the antigen-bearing microbes easily. Secondly, DCs have specialized receptors that help them do their job well. And number three, DCs, once activated, they migrate to area in the lymph nodes where they can easily encounter T cells. So that facilitates presentation. Number four, mature DCs express high levels of stuff you need to activate T cell mediated response, like peptide MHC complexes, co-stimulators or co-receptors, and cytokines. And number five, CDC1 transfers internalized proteins from phagosomes into the cytosol. So this matters because it helps the activation of CD8 positive T cells. Beside DCs, you have macrophages. What they do is presenting antigen that comes from a microbe that they have captured. And then the T cells will send them a kill command back to the macrophage to execute the captured microbe. So this is an example of what's called cell-mediated immune response. Another professional APC is B cell, who presents antigen to T helper cells. And that step is important for antibody production. And this is part of antibody-mediated response or humoral response. So those three are the professionals. So beside them, all nucleated cells also present antigens, but mostly to CD8 positive CTLs, cytotoxic T lymphocytes. They display the antigen on class 1 MHC. This is more for signaling to the T cells that there's something wrong in the nucleated cell. For example, the cell has been invaded by viruses. So the presentation of cytosolic antigens will call upon the CTLs to punch holes and kill the nucleated cells. MHC is designated differently between mice and humans. In mice, it's called H2 or histocompatibility 2, and in humans, we call it HLAs or human leukocyte antigens. So if you see HLAs in immunology literature, so that's just another name for MHC. In studying the MHCs, scholars have observed a phenomenon called MHC restriction. So that refers to the fact that CD8 positive T cell activation is restricted to antigens displayed by class 1 MHC, and CD4 positive T cells, the activation of it is restricted by class 2 MHC display. You can find class 1 MHC on almost every cells, actually, except erythrocytes or red blood cells. Class 2, you can find them on fewer cell types, such as dendritic cells, B cells, macrophages, and thymic epithelial cells. There's actually class 3 MHC as well. Maybe it'll come up in the future. Our cells deal with antigenic proteins differently based on whether it's cytosolic or ingested. For cytosolic proteins, which are the ones inside the cytosols, they are broken down by proteasomes and then displayed by class 1 MHC. For ingested proteins, which are proteins from the outside, they are degraded by lysosomes and then displayed on class 2 MHC. So again, for cytosolic proteins, they will be taken up by proteasomes and get degraded. The peptides will be transported into the endoplasmic reticulum through transporter protein called TAP. So in the ER, in the endoplasmic reticulum, the peptide, which is the antigen, it will bind to class 1 MHC and then transported and then displayed on the surface to activate CD8 positive T cells. For ingested extracellular protein, it will get sequestered from the rest of the cytosol first, and then the vesicle will merge with lysosomes, which will break the protein down. The pieces of the protein, the peptides, they will bind to class 2 MHC and then displayed on the surface to get the attention of CD4 positive T cells. So these are two MHC pathways for protein antigens. For non-protein antigens, T cells can still use MHC to detect small molecules and metal ions. Sometimes these small chemicals alter the shape of native peptides, and sometimes they alter the shape of MHC itself. So those alterations can trigger MHC-mediated T cell response. A subset of T cells can even recognize non-protein antigens without MHC, natural killer T cells and gamma delta T cells. NKT cells have the characters of both NK cells and T cells. So one way to recognize them is the presence of a receptor called alpha-beta T cell receptors. NKT cells can be activated by antigens in the forms of lipids and glycolipids, and these antigens are not displayed by MHC, but rather by a molecule called CD1. And lastly, gamma delta T cells. These cells have T cell receptors similar but not identical to ones that we know for CD8 positive and CD4 positive T cells. They have been shown to recognize proteins, lipids, phosphorylated molecules, and alkylamines, which can come from ammonia. We know that they don't use MHC, but we don't know what they use instead, or whether they need a display molecule at all. Okay, that's it. I know there's a lot going on in this episode. You have to rewatch it alongside the textbook a couple more times, but I think it will give you a strong foundation for later topics, especially the ones on adaptive immune response. So inshallah, I can go a bit faster on those ones and make the videos shorter. All right, keep on reading, keep on learning. Barakallahu li wa lakum. Assalamu alaikum wa rahmatullah.